1. Disfluencies and fillers. Real speech is littered with filler words that give speakers time to think. Examples. Like, well, so, right, yeah. Usage. Well, it's not exactly that simple. The data shows a different story. 2. False starts and restarts. People often start a sentence, change their mind, and restart. Since we cannot use ellipses, we use em-dashes to indicate an abrupt stop or change in thought. Usage. They found that, they realized the mutation was already there. 3. Active listening and interjections. In a podcast, the host won't sit silently during a long explanation. They will drop in small affirmations, reactions, or clarifying questions to show they are engaged. Usage. Expert speaker. The mortality rate in this specific cohort hit 61%. Host speaker. Wait, 61? That is, wow. Expert
Right.
speaker. Yeah, it's staggering. And it means we have to rethink everything. 4. Colloquialisms and contractions. Avoid overly formal phrasing unless deliberately making a scientific point. Use contractions heavily. Can't, that's usage. It's going to be a tough battle instead of it's going to be a difficult battle. 5. Overlaps and interruptions. While we use distinct speaker lines, the flow should imply that one person is jumping in right as the other finishes, or slightly before, indicated by conjunctions at the start of a turn or em-dashes. Usage. Host speaker. So you're saying the drug expert speaker, just bounces right off. Exactly. 6. Pacing. Break up long monologues. If a technical explanation requires several sentences, the other speaker should interject to anchor the listener. So if I'm understanding this right, the bacteria just totally changes its outer shell.
Yeah, exactly. It literally swaps out the building blocks. So when the drug...
The drug comes looking for the original target.
Right, the drug comes in, looks around, and just can't attach. It's totally useless. Natural notes.
Welcome to Outbreak Brief, the podcast that takes you inside the world's most dangerous infections. When you think of a truly terrifying pathogen, your mind probably jumps to, remote jungles or undiscovered caves or maybe exotic viruses jumping species in some crowded market. But today's target, doesn't lurk in the wilderness. It hides in the exact place you go to get healed, the hospital.
Yeah, it's really the last place you want a superbug setting up shop.
Exactly. So today, we're taking a deep dive into a microscopic villain known as Vancomycin-Resistant Enterococcus, or VRE for short. And what makes this pathogen so chilling is that it has essentially figured out how to use our own medical treatments against us. We are opening the medical files today to see exactly how this thing operates.
And to really understand the sheer scale of this threat, we have to look at what happens when it actually breaches a hospital ward. There's this recent, incredibly comprehensive 8-year study that came out of King Abdulaziz University Hospital in Saudi Arabia.
Right, the KAUH study.
Yes, exactly. So they tracked hospitalized patients who contracted this infection during that 8-year window, and the numbers are, they're grim. Out of those patients, an alarming 61.8% died.
Wait, 61.8%, that is... that's more than half.
It's massive, and it's not just an isolated regional issue either. If you look at the CDC data here in the United States, in just a single year, VRE caused an estimated 54,500 infections. And out of those, about 5,400 deaths.
Over 60% mortality in that ICU study, though. That number just completely shatters the illusion of the hospital as this, sterile sanctuary. It transforms the intensive care unit into a compromised safe house, like a place where the most vulnerable people are basically being picked off by an invisible predator.
That's a really accurate way to put it.
Which means we really need to figure out what kind of monster we're dealing with here. So what exactly is this pathogen? Let's build the profile.
Right, so let's start with the biology. Enterococci are gram-positive bacteria. To visualize that, imagine a cell with a single, very thick outer wall, kind of like a heavily fortified castle.
Okay, a microscopic castle. Got it.
But here is the truly unsettling part about VRE. These bacteria are not foreign invaders, at all. They are normally found peacefully coexisting right inside human intestines, the female genital tract, and even in secondary environments, like soil and water.
Hold on. So these bacteria are basically part of our normal, everyday microbial ecosystem. They live inside our guts right now.
They do, yeah. They actually help process food and maintain the gut environment. There are 2 main species we deal with in clinical settings. There's Enterococcus faecalis, which is the most common, and then Enterococcus faecium, which is the notoriously drug-resistant one.
Okay, so if they're naturally in us, why are they suddenly killing people in the ICU?
Because they don't always stay peaceful. The true villainy here, the thing that makes them a nightmare, is their resistance to a drug called vancomycin. For decades, vancomycin has been our heavy artillery, right? It's a powerful, last resort glycopeptide antibiotic.
The drug we use when everything else fails.
Exactly. It's this massive molecule designed to crush these bacteria when the standard drugs can't. And to understand why VRE is so dangerous, we kind of have to look at the microscopic mechanics of how the drug works.
Walk us through it.
So normally, vancomycin targets the bacteria's thick cell wall. It looks for a specific 5-piece building block, a pentapeptide called D-allodyla.
D-allodyla, okay.
Right. And when the antibiotic binds to this D-allodyla block, it sterically inhibits the bacteria. Basically, it physically blocks the bacteria from building its cell wall. And if the bacteria can't build its wall, the cell ruptures and dies.
Okay, I want to make sure I have a really clear picture of this because the biology feels almost sinister. The bacteria has this specific lock on its exterior, the D-allodyla block. And the antibiotic, the vancomycin, is a key perfectly designed to fit that lock, jam the door open, and kill the cell.
That's a great analogy, yeah.
Yeah.
But VRE has evolved. It literally changes the locks on its doors. The bacteria mutates that target receptor from D-allodyla to something called D-allodelac, or, sometimes D-allodecer.
Oh, wow. So the antibiotic key it no longer fits.
Doesn't fit at all. The vancomycin just bounces right off, and the bacteria goes on multiplying completely unharmed.
That is terrifying.
And the way it manages this lock swapping is fascinating. This resistance mechanism is encoded within operons. Think of operons as specific genetic cassettes or instruction manuals like the VAN-A and VAN-B genes.
Okay, so genetic instruction manuals.
Yeah. The bacteria keep this master blueprint on hand, which allows them to swap out those locks the second they sense the antibiotic in their environment.
But wait a minute. If this bug lived inside our own guts, completely harmlessly for most of human history, how did it suddenly get its hands on these genetic blueprints? Did we somehow force it to evolve into a killer?
Well, yes. We applied immense evolutionary pressure. We essentially created a survival of the fittest scenario on steroids.
How so?
Back in the mid-1980s, hospitals started heavily utilizing vancomycin and other broad-spectrum antibiotics to treat all sorts of various infections. Every time we flooded a patient's system with these powerful drugs, we killed off all the vulnerable, normal bacteria in their bodies.
Right, sweeping the board clean.
Exactly. And the only bacteria that survived that purge were the rare freak mutants that happened to carry these VAN-A and VAN-B genetic sets.
So by trying to wipe out infections, we basically cleared out all the competition for the one bug that couldn't be killed.
We did. And we didn't just clear the competition. We gave these survivors a permanent home. What the Saudi Arabia study captured so perfectly was this evolving epidemiology over time.
What did the data show?
It showed that strains carrying these resistance genes used to be mere colonizers, just harmless hitchhikers sitting quietly in a patient's gut. But over time, with repeated antibiotic exposure, they transitioned. They crossed the line from silent passengers into aggressive pathogens, causing these deadly clinical infections.
Okay, that makes sense for how we bred this invincible survivor inside a single patient. But for it to become a global hospital crisis, it has to escape that host, right? It has to find its next target. How is it jumping from a colonized gut to the person in the next ICU bed?
We have to be really, really clear about transmission routes here. VRE is not airborne. It does not spread through coughing or sneezing.
So you can't catch it just by breathing the air in a hospital room?
No, not at all. It spreads entirely through direct contact. The primary vehicles are contaminated environmental surfaces, shared medical equipment, and, unfortunately, the unwashed hands of healthcare workers moving from room to room. It can even transfer from the GI tracts of animals into aquatic environments. But in a hospital, it's mostly surfaces and hands.
I have to push back on that a bit, though, because if it's not airborne, shouldn't it be incredibly easy to contain?
You would think so, right?
Yeah, I mean, if there's no coughing involved, standard ICU wipe-downs and hand washing should stop this dead in its tracks. Yeah. Why is it still spreading like wildfire if it's just sitting on a bed rail?
The difficulty of containment really comes down to the chaotic reality of critical care and the sheer biological resilience of this bacteria.
Resilient how?
Well, yes, hand hygiene and surface cleaning are effective, but Enterococci are famously hardy. They can survive on dry surfaces, I'm talking bed rails, doorknobs, IV poles, heart monitors, for days and sometimes even weeks.
Weeks, just sitting there on dry plastic.
Just sitting there waiting. And think about an ICU. Healthcare workers are performing dozens of rapid high-stress interventions every single hour. A nurse might silence a monitor, adjust an IV line, and then reposition a patient's blanket.
All in the span of 30 seconds.
Exactly. A single microscopic lapse in that rapid-fire chain of contact is all it takes to move the bacteria from the plastic to the patient. And the reason it's spreading so effectively is that the patients it targets are essentially defenseless.
Let's talk about those patients,
Mm-hmm.
because that is crucial. Who is in the crosshairs here?
The data from the King Abdulaziz University Hospital study paints a really vivid, tragic picture of the vulnerable population. The median age of an infected patient is 61. It aggressively targets those with severe underlying health conditions, people whose immune systems are already just completely exhausted.
What kind of conditions are we talking about?
In the study, 63.4% of infected patients had diabetes, 62.2% had hypertension, 52.4% had chronic kidney disease, and over 26% were malignancy or chemotherapy patients.
Wow, so we are talking about severely immunocompromised individuals who are literally already fighting for their lives.
And the medical interventions designed to save their lives actually dramatically increase their risk of getting VRE.
How does that work?
Prolonged hospitalizations, complex surgeries, and especially the use of indwelling medical devices, things like urinary catheters, central venous lines placed in the neck or chest, and endotracheal tubes for ventilators.
It's a tragic irony. A patient goes into the ICU for heart failure. The doctors put in a central line and a catheter to help them deliver medications, monitor fluids. Then they give them broad-spectrum antibiotics to protect from generalized hospital infections.
Right.
But that exact combination acts like a Trojan horse scenario. The antibiotics nuke the normal, helpful bacteria, clearing the battlefield, and those plastic tubes provide a direct, unguarded highway straight past the skin and into the body.
You've just described the exact mechanism of a modern hospital-acquired infection. It's a perfect storm. The study noted that initial colonization upon ICU admission is a huge factor, too.
Meaning they bring it in with them.
Yes. Up to 10% of patients arrive at the ICU already harboring VRE in their gut from previous healthcare exposures. And then another 10% will actually acquire it during their current stay. Once it breaches those physical defenses like the skin or the urinary tract through a catheter, the disease progression is just brutal.
So walk us through the sensory reality of this. If these bacteria are setting up camp in IV lines and catheters, what happens when it actually strikes? What does the patient experience?
Well, VRE is kind of a shapeshifter. The symptoms depend entirely on where the bacteria lands and sets up camp. Based on the clinical data, 46.5% of cases present as urinary tract infections, mostly due to those catheters.
So that would mean burning pain, high fever, pelvic discomfort.
Exactly. But when it enters the blood, it becomes catastrophic. It causes devastating bloodstream infections, which we call bacteremia, in about 18.9% of cases.
And bacteremia isn't just a local infection with a fever, right? That's systemic.
It is systemic shock. The immune system detects the bacteria rapidly multiplying in the blood and just goes into total overdrive. The patient experiences sudden, violent chills. Their blood pressure plummets, cutting off oxygen to vital organs, which leads to multi-organ failure.
Oh, that's horrific.
It gets worse. VRE also causes infected surgical wounds that refuse to heal, turning red, swollen, and necrotic in about 9.5% of cases. It can cause intra-abdominal infections and, worst of all, endocarditis.
Endocarditis, that's the heart, right?
Yeah, it's a lethal infection where the bacteria actually colonize the heart valves themselves, literally destroying the tissue with every heartbeat.
But logistically, this sounds like an absolute nightmare for the doctors. I mean, think about it. A patient is on a ventilator for respiratory failure or on dialysis for kidney failure. They already have a dozen reasons to spike a fever or have their blood pressure drop. How do doctors even know if a sudden fever is from their underlying disease or if VRE has crashed the party?
They don't, at least not right away. That diagnostic confusion is the daily grinding reality of the ICU. You can't just look at a patient and diagnose VRE. To confirm it, doctors have to draw blood, take urine samples, or swab wounds and send them down to the lab.
Right.
And this brings us to the truly harrowing reality of trying to treat the superbug once you finally spot it.
Because, as you said earlier, our best weapon, vancomycin, is completely off the table. It bounces right off.
It's actually worse than that. Over half of the pathogenic isolates of Enterococcus faecium are intrinsically resistant to other frontline drugs as well. Things like ampicillin and high-level aminoglycoside.
Wait, half of them resist the backup drugs too. So how do you treat a bug that just eats antibiotics for breakfast?
You have to systematically test everything you have left. Doctors rely on antimicrobial susceptibility testing, or AST panels. Hospitals use these automated lab systems like the VTech2 system.
How does that work?
They take the bacterial specimen from the patient, isolate it in the lab, and expose it to a panel of alternative, highly specialized antibiotics. Drugs with intense side effects, like daptomycin, linezolid, teicoplanin, or tigecycline, they're basically checking to see if any of these backups can still breach the bacteria's mutated defenses.
Okay, but how long does that VTEC2 system take to give the doctors an answer?
It takes anywhere from 10 to 18 hours just to generate those susceptibility results.
You're kidding. 10 to 18 hours. You're telling me we have to swab, culture, and run autonomous lab tests that take nearly a day just to pick the right drug?
I am.
But if a patient is in septic shock from a bloodstream infection, aren't doctors racing against a clock with their hands tied? Their organs are starving for oxygen right now. You can't just wait until tomorrow afternoon to see what drug works.
You can't. But the reason it takes that long is that you aren't just running a quick chemical scan. You are waiting for biology to happen. The lab literally has to grow the bacteria in an incubator alongside the different antibiotics and watch in real time whether the bacteria survives or dies. You cannot fast forward life.
So what do the doctors do in the meantime? Just watch the patient.
No, they guess empirically. While the lab is waiting, the doctors are forced to prescribe a cocktail of powerful drugs, hoping one works while the clock ticks down. If they guess wrong, the bacteria continues to multiply. Time is tissue, and in bacteremia, time is life.
With treatment being this delayed and frankly this difficult, it makes you wonder about alternative defenses. Why don't we just vaccinate vulnerable populations against VRE before they ever get admitted to the ICU?
Well, currently there is no vaccine available for VRE at all, and relying on natural immunity is essentially moot.
Wait, I think I see the problem here. Earlier you said the Enterococci genus is part of our normal gut flora?
Yes, exactly.
So if we created a vaccine to hunt down and kill VRE, wouldn't we accidentally trigger the immune system to attack our own healthy, functioning gut bacteria? We'd basically be inducing an autoimmune crisis.
You've hit on the exact biological hurdle. You cannot easily train the immune system to eradicate a bacterium that shares so much genetic architecture with the beneficial microbes keeping your digestion running.
That makes perfect sense, but it's totally discouraging. So what is in the research pipeline? Are we just out of luck?
Because traditional vaccines aren't the answer, researchers are looking at radical alternatives mentioned in the recent literature. Things like antimicrobial peptides, and most fascinatingly, bacteriophage therapy.
Bacteriophages. Okay, those are the viruses that look like little microscopic lunar landers, right? The ones on those electron microscope images.
Yes. Bacteriophages are viruses that naturally hunt and eat specific bacteria. So instead of dropping an atomic bomb of broad-spectrum antibiotics that wipes out the whole gut microbiome, a phage acts like a biological sniper rifle.
So that's clever.
It lands on the specific VRE bacteria, injects its viral DNA, and forces the bacteria to produce more viruses until it literally bursts from the inside out.
Wow.
It's brilliant, but it is still highly experimental and definitely not yet widely available in standard ICUs.
So if our pharmaceutical defenses are that limited and vaccines are totally off the table, it sounds like humanity's primary weapon against this thing is just extreme behavioral and environmental control.
Prevention is everything. Right now, core infection control practices are the only reliable front line we have. We are talking rigorous hand hygiene with soap, water, and alcohol sanitizers.
The basics.
The basics, but executed perfectly. Strict personal protective equipment, gloves, and gowns for any contact with body fluids. Daily, meticulous room cleaning with industrial disinfectants. And in high-risk areas, hospitals are implementing rapid VRE molecular screening.
Is that faster than the 18-hour wait?
Much faster. Technologies like the GenExpert system can detect the VAN A and VAN B resistance genes directly from a swab in about 2 hours, completely bypassing that terrifying 18-hour culture wait.
2 hours changes the game completely. It gives doctors a fighting chance to isolate the patient before they spread it to the rest of the ward.
It does. And speaking of isolation and environmental control, there was a fascinating anomaly in that Saudi Arabia study that perfectly illustrates how transmission actually works in the real world.
What was the anomaly?
Well, the researchers mapped out the infections over the whole 8 years, right? VRE cases peaked heavily in 2021.
2021, the height of the COVID-19 pandemic.
Exactly. COVID overwhelmed the healthcare system. The sheer volume of patients, the physical and emotional exhaustion of the staff, and the severe shortages of PPE caused temporary lapses in those meticulous infection control measures. Chaos basically bred contamination.
That makes total sense, sadly. When the ICU is overflowing and alarms are blaring, a nurse might not have the time to perfectly sanitize every single inch of an IV pole before rushing to the next coding patient.
Exactly. But then, in 2022, VRE cases in that same hospital absolutely plummeted, and the mechanics of why are incredible.
Wait, why did they plummet? Did they get a new drug?
No. The extreme self-isolation, the strict hospital lockdown protocols, the restriction of visitors, and the severe reduction in patient mobility that were implemented to fight COVID-19. Well, they accidentally crushed the spread of VRE in the hospital.
That is wild.
By keeping everyone locked in single, isolated rooms to prevent a respiratory virus from spreading through the air, they inadvertently severed the contact transmission lines of this bacterial pathogen on the ground.
It's an amazing paradox, isn't it? I mean, the extreme, exhausting measures we took to fight a viral respiratory pandemic accidentally became the perfect net to catch a bacterial surface epidemic.
It really is.
It proves that extreme isolation works mechanically, but it also shows how impossibly resource-intensive it is to actually stop this bug on a normal day. Which brings us to the global landscape. Where do we stand right now with VRE?
Right now, VRE is firmly endemic in hospitals worldwide. It is a permanent resident. The predominant species globally is now Enterococcus faecium, and the highly resistant VAN-A phenotype makes up over 40% of the genotyped samples in recent studies.
40%. So almost half are the absolute worst-case scenario.
Yeah. The CDC is actively tracking VRE through the National Healthcare Safety Network, monitoring the data on their patient safety portal. Health departments and federal agencies are basically fighting a localized trench war, bed by bed, in ICUs across the globe.
Endemic. Wow. It sounds like we've reached a really tense stalemate. We've accepted this superbug as a permanent, terrifying resident of our medical system, and now we're just trying to manage the numbers and limit the casualties rather than actually eradicating it.
That is the grim reality of the present. And looking to the future, this pathogen remains highly unpredictable. The biggest remaining unknowns revolve around its genetic evolution.
Is it still changing?
Constantly. In the recent surveillance, researchers noted growing instances of genotype-phenotype incongruence.
Hold on. Genotype-phenotype incongruence. What does that mean in plain English for the doctors trying to treat it?
It means the bacteria is wearing a disguise. A bacteria might possess the VAN-A gene, which usually gives it a specific type of extreme resistance, but it expresses itself physically like a VAN-B phenotype.
Oh, so it's evolving in ways that trick our tests.
Yes, it confounds our standard molecular testing, making it even harder to choose the right backup drug. Future preparedness relies heavily on one crucial concept if we want to survive this: antimicrobial stewardship.
Meaning we have to stop handing out antibiotics like candy.
Exactly. We must stop overprescribing broad-spectrum antibiotics, specifically things like carbapenems and cephalosporins, in both human medicine and agriculture. Over 50% of the patients in the Saudi study had recent exposure to carbapenems and vancomycin.
So the doctors were priming the patients for VRE before they even got it.
That unnecessary, heavy-handed usage is the exact evolutionary pressure that breeds VRE in the first place. We have to be more disciplined.
You know, it really makes you think. If our strongest weapons, our broad-spectrum antibiotics, are the exact things training these bacteria to become invincible, we aren't just fighting a pathogen anymore. We're fighting the consequences of our own medical advancements.
That's the scariest part of all this.
It is. Our attempts to create a sterile, safe environment for the sick ended up breeding a predator that thrives exclusively inside it. It really forces you to wonder, if we keep applying this evolutionary pressure, what does the world look like the day the very last antibiotic fails? Stay informed. Stay prepared. This has been Outbreak Brief.